Hello, everyone. My name is Daniela Portela, and I am a second-year behavioral neuroscience student, and I plan to graduate in the spring of 2024. For my co-op CaNCURE project, I worked on the sustained delivery of Talazoparib for the treatment of BRCA1 deficient ovarian cancer. I was really grateful to work with Professor Shri. I learned a lot in the lab, similar to things about cell culture, which I had never done before, working in sterile environments, and learning a lot about ovarian cancer. I'm going to go through a little bit about what my research was on, more specifically. So, ovarian cancer is one of the deadliest cancers of the female reproductive organs, in addition to being the fifth leading cause of cancer deaths among women in the United States. There are many causes to ovarian cancer, including those being linked to age, genes, and hormones that one's body produces. The three main types include germ cell, struggle, and epithelial. My project specifically focused on BRCA1 deficient ovarian cancer. And the problem with ovarian cancer, what makes it so deadly, is that it's very hard to diagnose, specifically since the symptoms are not prevalent. So I included some statistics. About 19,880 women will receive a new diagnosis of ovarian cancer, and about 12,810 will die from ovarian cancer. There are some treatments that exist now. I worked with a type that of PARP inhibitor that is known as Talazoparib. So as time goes by, we develop more and more methods for early detection, but our main focus should also be on improving the mechanisms we have now. PARP inhibitors are used to treat women with advanced BRCA deficient ovarian cancers. The way that PARP inhibitors work is that they prevent the repair of DNA single-stranded breaks and promote the conversion of single-stranded breaks to double-stranded. Therefore, PARP inhibitors, the one I worked with specifically was Talazoparib, PARP inhibitors can regulate the function of proteins involved in the repair of DNA, so therefore it can modulate the DNA binding activity of BRCA1. BRCA1 itself is a cancer gene that produces proteins that help repair damaged DNA, which essentially lead to cancer and tumor growth. I specifically worked on sustained delivery of TLZ. So the purpose of why the implants were even made is for the sustained delivery to occur. And the sustained delivery of the implant also reduces toxicity compared to taking the drug orally. So for my methods, Sridhar lab created the Talazoparib implant using PLGA, and the implants were made with solvent extraction method. We checked the loading of the HPLC, which shows the success rate of the sustained delivery. And the aim of the project is to see just how effective PARP is on killing tumor cells and how smaller the colonies get as the cells are treated with this drug. So for cell culture, basically the 366 cell line, which is what I used, those cells were frozen, and we had materials gathered such as trypsin, PBS, and 366 media. They were heated up in a water bath. Cell culture would be performed by me, and then I would either turn into a new flask or colony genesis. Colony genesis was important because this is where we got the most results from. We counted a specific number of cells and treated them with treatment. And a new flask was placed just in case the flask that was already there was confluent, so there was a lot of cell growth. As the results for colony genesis, you can see that the in-cell TLZ killing effect had a great killing effect in the two TLZ, the 1 and the 2. Cell viability was 100 before any treatment was placed, and then it decreased to 10 when the treatment was introduced. So to the right, you can see the plate, the control, and the TLZ implant, and the blank. You can see how little growth there is in the TLZ column. So IC50 basically tells us how much drug is needed to kill 50%. And I use this by cell viability. It was done to find the IC50 of TLZ. It also provides a measurement of this drug and how effective the drug is to cancer cells. To the bottom right, I provided an equation, and you can see that the IC50 was 14.2735, which is very low, meaning that the cells are very sensitive to the drug. And since it can be effective at a lower dose, it reduces toxicity for the patient. So to conclude, Talazoparib kills BRCA ovarian cell lines. The tumor cells treated with TLZ implants showed decrease in colony number. And we achieved an IC50 that showed it is highly sensitive to the therapeutic. Also, it was important to note that keeping cells sterile is necessary to prevent contamination. We experienced many contaminations, and the problem with that is that we would need to throw out all of the materials that we had used. And it's super important to sterilize every equipment with ethanol. As for future steps for sustained delivery, this would really make the lives of the patients much easier without needing to go to the hospital as frequently as they do to get chemo. As for PARP inhibitors, PARP success rate in inhibiting replication of damaged DNA cells could prove to be effective in other types of cancer. The TLZ spacer, the next step for the spacer, is that it would need to be approved by the FDA. As for CaNCURE, as I mentioned, the CaNCURE experience has taught me a lot about working with live cells, and I'm definitely interested to see just how far we get in cancer research. I want to thank everyone who's a part of the CaNCURE co-op and the NCI grant. I would also like to acknowledge the staff, my mentor, Professor Shree, my day-to-day mentor, Shi-Cheng Yang, for their guidance and support, and additionally Anne Van Maloney and Nita Brown for the help throughout this CaNCURE co-op. Thank you.